We are here in Smithali for the sludge gas sample collection. We will show the entire process of fecal sludge sampling, its transportation to ENPHO lab, storage and lab preparation. We'll cover each step carefully and systematically. The sampling site is located in Smithali within Ward number 8 of Mahalakshmi municipality, approximately 2 kilometers south of Lubu Bus Park. The containment unit stores black wastewater generated by a household of 5 members. It was constructed in 2009 and was last emptied approximately 4 years ago. It consists of a simple pit structure with a semi-permeable side walls measuring 36 inches in diameter and 10 feet in depth. There is no signs of overflow or leakage has been observed in the surrounding area, indicating that the containment is functioning adequately. The pit is accessed through a manhole opening that is secured with a protective slab. The researcher begins by arranging all the equipment and materials needed for sampling, such as core sampler, sample container, measuring tape, permanent marker, ice box, tissue paper, and disinfectant spray. Before getting started, researcher puts on personal protective gear such as gloves, rubber boots, masks, waterproof apron, and protective goggles. This ensures safety against harmful gases and pathogens. Next, the researcher prepares core sampler by fitting a metallic rod with an airtight seal in the bottom and attaching a rope at the top inside a PVC pipe of 50 mm diameter. Researcher removes the cover slab of the pit and observes the sludge inside. To determine its depth, researcher carefully lowers a long stick into the containment and then withdraws. The sludge leaves a clear mark on the stick, which is then measured with a measuring tape. The distance from the base of the stick to the mark is measured to be 5 feet, which is the depth of the sludge. A sample is collected from this level to ensure it is truly representative. Researcher holds the core sampler device and lowers it through the manhole to the bottom of the pit. After letting it settle for about 30 seconds, the sampler is sealed and removed. The collected sludge is then transferred into a bucket. This process is repeated until the bucket is full. Once enough sludge is collected, the researcher homogenizes the sample by mixing it thoroughly. The sludge is transferred into a 10-liter container and a portion of the sample is poured into a 1-liter transparent measuring cylinder and allowed to settle. The same procedure is carried out again to collect the next 10-liter sample. Type of fecal sludge is also determined by calculating the volume of the fecal sludge settled in the measuring cylinder. In this case, the fecal sludge type is determined to be semi-solid and the color is noted to be black. Finally, once all samples are collected, researcher carefully cleans and disinfects all the equipment, ensuring everything is ready for safe storage or transport. After collecting the sample, the ice box along with other necessary materials are loaded into the vehicle for transfer to the laboratory. The lab is located about 12 km from the sampling site and it takes around 30 minutes to reach lab. Upon reaching the ENPHO lab, researcher removes the sample containers from the ice box. The sludge is transferred into a 20-liter bucket and mixed thoroughly using a stainless steel ladle to ensure homogeneity. The mixed sample is then divided into 3 parts or triplicates. For the storage of the sample, researcher arranges the different volume of sample bottle based on different type of analysis. For the physiochemical parameters such as total solid, pH, TSS, turbidity, pH is the ammonia, sludge volume index, orthophosphate, nitrite. Researcher stores the sample in 1-liter bottles without preservatives. pH and conductivity are analyzed immediately, while the remaining parameters are tested later after refrigeration. For the parameters such as COD, TKN, and total phosphorus, samples are preserved with sulfuric acid to keep the pH below 2. And for microbial analysis, researcher stores the sludge sample in the sterilized containers. Throughout the entire process, standard sampling protocols are strictly followed. with careful handling at every stage to ensure accuracy and reliability of the results. This completes the process of fecal loss sampling, transport, and storage for laboratory analysis.